and provides comprehensive care for the many complications of this condition. In collaboration with cardiologists and respiratory physicians, she's developed a multidisciplinary team for assessment of cardiopulmonary complications in scleroderma at the hospital. Wendy's a founding member of the Australian Scleroderma Interest Group, and this group of physicians with an interest in scleroderma was formed in late 2006. The initial project of the group was to develop a national screening program for cardiopulmonary complications of scleroderma, and she's played a major role in the development of this program and the Australian Scleroderma cohort study and the development of a national scleroderma database. The National Scleroderma Database records annual comprehensive data on patients with scleroderma. Since 2007, she's taken a major role in the day-to-day management of this database and analysis of the data from the database. The major focuses of the research to date have been cardiopulmonary complications of the disease and the development of systemic sclerosis damage index, and more recently, an activity index. She's participated in many clinical trials in scleroderma and PAH. And it gives me great pleasure to welcome you, Dr. Wendy.
Thank you. So I will now share my screen. Share my entire screen, I think would be best. Whoops. And now I need to put that on. It won't let me share it. Share a window. Okay, so that's what we'll share, a window.
Yes.
Yes, that's better. All right, now I'll put it in speaker mode, so I can't see you, but can everyone see this? You're presenting. Okay, click here to return to the video call. Okay, so I just want to put it in presenter mode so you don't see all the other slides. Okay, is that okay? Can you see all that?
Yes, perfect. Thanks, Wendy.
Beautiful. All right, so today I'm talking about clinical trials. I think the poll's really interesting, and I know what the unmet need is. But what we're talking about here really today is, and I'll talk about some other clinical trials very briefly at the end, but predominantly today I was given the brief of talking about the sort of pharmaceutical development of new drugs that we might see for scleroderma. All right. So let's just go back to the beginning. So what sort of types of research occurs? So this is just a sort of basic sort of run through of research in scleroderma and research in any disease. So there's all types of research we need in scleroderma. We need to, on the left, understand the basic science. We need to know what's going wrong in the body. And constantly we're understanding more about the immune system and the cells and how cells interact. And that's not just in this disease, it's in many other diseases as well. And so, you know, a lot of work is being done, not so much in Australia because we're not really set up for that, but there's a lot of basic science going on into fibrosis, and not just fibrosis in scleroderma, but there's other conditions where there's excessive fibrosis, like excessive fibrosis in people with idiopathic interstitial lung disease. So interstitial lung disease like we see in scleroderma, but they haven't got scleroderma. We also see excessive fibrosis in diabetics in their kidneys that causes long-term damage to their kidneys. So everything's different, every scenario is different, but there are some basic overlaps. So there's a lot of basic science going on in the world. Basic science costs billions of dollars, and you've got to have the right people set up for it. So people often say, oh, why aren't we doing it in Australia? Well, we just don't have the money, we don't have the expertise, but that doesn't mean in the community we're not sharing it all the time. You know, when we go off to meetings, it's because we're learning from each other, we're hearing what's going on. That's why we read journals for entertainment at night. And there's also animal models of disease. People in all diseases try and create an animal model so that you can test drugs, test what's going on. They're never quite the same as the disease. The other thing, and the thing that we do a lot of in Australia, is the epidemiology. That means the study of the disease. It means the patterns of disease. We all know everyone with scleroderma is different. Who gets bad lung disease? Who gets bad skin disease? How can we predict the outcome? How can we predict the people who are going to do badly, the people who are going to do well, the people who do need treatment, the people who don't need treatment, you know, and what the future holds for people, how active their disease is. So actually the Australian Scleroderma Cohort Program database has really been world leading in some of this epidemiology that teaches us about the patterns of disease, that's taught us about how to predict outcomes, that, you know, is involved in new screening tools. And then on the other side, you can see clinical trials. And the sort of clinical trials today, I'm going to talk a lot about new therapies and new treatments. But, you know, there are other types of clinical trials that we'll talk about. And when we talk about clinical trials, there's a variety of things you can do. The first of it is to try a new therapy. You've worked out that this particular cell or this particular enzyme or chemical is turned on in this process of fibrosis or in immune cell activation. And then you've found a drug that blocks that molecule, and you might have tested it in the lab, and then you might have tested it in animal, and then you move into testing it in people. And then the other thing is sometimes you've got a drug that's already approved for some other disease and you think, well, wonder if it would work in scleroderma. And that's another thing that often happens, is that a drug that's well used is then gets, for another disease, gets tested in scleroderma. So it is a complex thing. It's not a simple process. And it's a bit of a circular thing. You've got to understand the disease itself, and that'll become quite obvious today that you need to understand the patterns and the risk factors for getting worse, to choose the right part of the disease, the right time to target the disease. But we also need to understand how to measure things. How do we measure that the disease is getting worse? We need hard endpoints to measure. We can't just go to governments and say, I want you to spend a few $1 billion on this drug because we think it works, or people feel better on it. They need hard endpoints. They need something to say, this is what happens if you use this drug. So sometimes we have to work on measures of actual disease progression, you know, trying to choose which of these markers shows that people are getting worse, or they're going to end up with damage, or they're going to end up with lung failure or bad skin. And then we're often looking for biomarkers, blood tests, simple things that are harder, things that are a little bit, you know, like they've definitely got this in the blood and this is what predicts that. So that's what a biomarker is. And then we also need to understand, as I say, the cellular level, but it all feeds into each other. So there's lots of research that's gone on. The development of the damage index and the activity index, where the team at St Vincent's Melbourne, our research fellows, have led that activity and damage index, might be ways that in clinical trials that we can show drugs are working, because it's not easy in scleroderma. And one of the reasons it's not easy in scleroderma is it's such a complex disease. If you just all put your screens on and look at each other, you all look a bit different, and you've all got different types of disease. And when you talk to each other, everyone's got different problems. Some people's problems are the tight skin, and that's this group of patients with diffuse scleroderma over here on the left of the screen. Some people's problem is more vascular. It's the digital ulcers. It's the severe Raynaud's. It's the narrow blood vessels in the lungs that can cause pulmonary hypertension. Some people's problem are stiffness in their joints or inflammation in their joints. Some people get kidney problems, some people don't. Lots of people get gut problems. But again, that's incredibly variable. So it's this incredibly variable disease, and there's lots of different pathways involved. Here, where we've got the bad skin thickness, we know there's lots of increased collagen in the skin. A drug that turns off that might not help the person who's got problems in their fingers with ulcers, because that's probably more a small blood vessel problem. It's probably not that we'll ever find one drug that turns off everything. And as I say, we're saying, you know, it really is a sort of three processes. There's inflammation that can cause inflammation in muscles and joints. We see that most patients have autoantibodies. That's part of the autoimmunity. But we also see this blood vessel problem where the blood vessels are very narrowed, and that results in the Raynaud's or the digital ulcers or these nail fold changes. And also there's the fibrosis, too much collagen, too much scar in the lungs, thick, tight fingers, thick, tight skin everywhere. And again, as I've just said, everyone's different. Some of you will have interstitial lung disease. Some might have narrow blood vessels in your lung, pulmonary hypertension. Some might have gut involvement. Some might have nothing, just tight skin. Some might just have a few telangiectasia and a little bit of tight skin. Some of you might have muscle. Many of you will have fatigue. But everyone's different. And this is where it is important when we're looking at trials to understand what the trial's about and why everyone's not included. Because if we're looking at a trial for interstitial lung disease where we think it might change the fibrosis in the lung, that's the group of patients we want. When we go back to what's going on in the body, we know it's complex. And this is a really simple, it may not look simple, but it is a simple scheme. And we think there's a whole lot of things that might trigger it at the start. And we don't exactly know everything that's going on. What we know is that there may be some injury to the system. That might be in the blood vessels, that might be damage to the lining of the blood vessels, that might lead to activation of platelets, activation of the blood vessel wall that lets various chemicals that are normally in the body system move into the tissue space. It may be the immune system is upregulated, and as I say, nearly everyone has got an autoantibody, an anti-nuclear antibody, which suggests that the immune system is upregulated. And there are so many parts of the immune system, and we're learning more and more. And then we also know that all of these chemicals, and everyone's different. Not everyone has got elevated endothelin-1. Not everyone's got elevated TGF-beta or IL-6. That results in activation of the fibroblasts. The fibroblasts are the normal cells that we all have in our bodies that make collagen, collagen being the structural protein that sits between cells. We all need collagen. If we don't have any collagen, our skin won't hold together. But what we know is that in people with scleroderma who've got a lot of thick skin, they've got activation of these fibroblasts, there's more myofibroblasts, there's more collagen, there's remodeling, there's scarring, but there's also the immune system in it. And every single person has some different combination of this. And we call it all scleroderma or systemic sclerosis, but probably in each person there's different combinations going on. And this again is the huge challenge of scleroderma. So I've just harped on for a few minutes saying scleroderma is different in every person. We classify scleroderma into two major subsets. Those major subsets are called limited scleroderma and diffuse scleroderma. That's based on the extent of your skin involvement. There are some differences in how people present with scleroderma with limited and diffuse, and there are some differences in the internal organ involvement. So limited scleroderma normally begins slowly. When patients come along to see us as a rheumatologist, they've often had symptoms for years. They've had Raynaud's that's come on for years. It might have got worse over the years. They've often had puffy fingers, slightly tight skin or thickened skin on their fingers for a long time. They may have developed some telangiectasia or red dots, but the skin involvement, the thickening of their skin, not the pigmentation, the thickening and tightness of their skin is always limited to below their elbows down to their fingers, below their knees down to their feet, on the face and the front of the chest. But it's generally a disease that comes on slowly, and you know, internal organ involvement can occur. The gut's often bad in limited scleroderma, as is the Raynaud's and the calcinosis, but the skin generally doesn't progress to the diffuse tight skin everywhere, and often the skin involvement's quite mild. In diffuse scleroderma, people present rapidly. Their Raynaud's started in the last few months, and very rapidly often they're initially diagnosed with rheumatoid arthritis or lupus or something because they have acute joint pain, very swollen feet and hands, and then very rapidly over 6-12 months, they have explosive involvement of their skin with tight skin either on their upper arms or their thighs, on their tummy. And to be classified as diffuse, you've had to have had it in that area at some stage. Again, this is a group of people that often, and it only makes up 20 to 30% of scleroderma in Australia. About 70% of scleroderma in Australia is limited scleroderma, but diffuse scleroderma is the group that do sometimes get fairly rapid internal organ involvement. But there are people with bad lungs who've got limited scleroderma. And this is an important graph we might come back to later. And it shows what happens to the skin disease in scleroderma. And it's one of the important things in designing clinical trials is that the skin rapidly gets worse over the first 1 to 2 years in diffuse scleroderma. It spreads rapidly onto the upper arms and thighs. And then it often plateaus off. And then it often really drops down. And even 10, 15 years later, sometimes people have still got curly, tight fingers, but their skin is very thin. In limited scleroderma, it might gradually progress a tiny bit over the years, but not much. So if you've got a drug that you think is going to stop the skin progressing, it's going to be important that you choose the right group of patients. If I choose a whole group of patients with limited scleroderma, and over a year or so, their skin doesn't progress, I'm going to say, great job, great drug. Look, it worked. But they weren't going to. Their natural history was that they weren't going to get worse. If I choose a group of patients with diffuse scleroderma who are 5-10 years out and their skin starts improving and I give you my drug, I'll say, great job, my drug works. But actually, this was the natural history. So we've often got to, if we've got a drug that we think helps skin stop getting worse in diffuse scleroderma, we need to get those early patients in the first 1 or 2 years. And that's actually quite hard to do because by the time they get to specialist centres, they're often 1 or 2 years out. But it is the importance of choosing the right patients. Early on, we didn't understand that. There's a trial of, something we never use now called penicillamine that we know doesn't work now, but the early trials used lots of late and limited, and they said, great, look, it works, and they didn't have it placebo-controlled. So this is the importance and why trials are often restricted to a very small number of patients with early scleroderma if skin's the outcome. That doesn't mean later, when we've proven that the drug works, it might not be useful for other groups. But to prove it works, we've got to make sure we choose the right patients. So I guess getting back to what we do with basic science has showed us that it's really complicated. Everything is just interrelated. It's not simple. People often say, oh, I've read this paper about such and such doing such and such, but it doesn't always work when we put it into real people. So the pathways to a new treatment is to start with basic science. It's to look at what might regulate normal fibroblasts, or upregulated fibroblasts taken from skin biopsies of people with scleroderma. And then they put those fibroblasts in a test tube or a tissue culture, they grow them, and then they play with chemicals and the immune system and things. So we might have found a new pathway in the fibroblasts that seemed to be, involved in these activated fibroblasts in scleroderma. But what goes on the test tube isn't real life because there's all these other pathways that are modulating that. There's all these feedback loops. It's important to understand what happens when we block that pathway. Does something happen? It seems to be upregulated in these fibroblasts, but when we block that pathway, does something happen? Doesn't always, because you've got other chemicals. And what are the harmful effects of blocking that pathway? Often that pathway is occurring in other cells as well. And if we block that pathway, sometimes other cells die or something bad happens. And then after you've, played around with the lab and you think it's reasonable and you think it's okay and you think it's doing something, but not too much, it's not killing off all the cells, then they generally model it in animal models. And there are a few models of animal models of scleroderma. And then eventually it's tried in people. So how do you develop a new drug? It's complex. It's a hugely long process. It costs billions of dollars, and that's why most of this starts with basic science in basic labs and then eventually gets translated by big pharma. They need to take it on. We can't take this on ourselves. As I've said, you start in the lab, you find a chemical, you test the effects in the lab, and then you test it in animals. But you have to be very careful about animal models because not all animals are the same as us. We've all got differences. Even in mammalian animals, we've got different pathways. We metabolize things differently. That is, we break down the drugs in a different way. And all animals have different pathways. So you've got to choose the right animal model. And then these sort of studies and very careful studies in animal models, they give it to rats and things. And I know that sometimes is a little confronting, but we need these to understand the pathways and to understand what badness can occur from you altering this pathway, because pathways are in there for a reason. Yes, they might be overactive in scleroderma, but if we completely stop it, what's going to happen? And we also need to know how the drug's broken down and how's it excreted by the body. So, how long does it take to get out of the body? Is it broken down by the liver? Or is it broken down by the kidneys? Or is it excreted in the gut? Because everything you put in comes out some way. It's generally changed. And you also have to understand if those changed products in metabolism cause problems. And sometimes you have a pathway that you think's great, and then they put it in an animal model and they find that there's all these off-target effects that they couldn't have predicted. And, by the time we get one drug out, there's been truckloads of that, billions and billions and billions of little other compounds that have not got that far. So it's not simple. So what's the next step? Once you've tried it in animal models, the next step is to see if it works in a scleroderma animal model, and the classic ones of the Blair mouse and lung model, which is a fibrotic lung model, the tight skin mouse, which is a particular genetic variant of mouse, and then tissue cultures. And as I've said, again, altering one pathway might stop something we need. The next step is to move it towards human trials. And there are 4 phases of clinical trials. So trials are always labelled by the phase. You might see if you look up trials, you'll see phase 1, 2, 3, and 4. Phase 1 is the first time we give the substance to a human. And unless it's a cancer model, normally these are normal healthy volunteers. There are people out there that volunteer for these studies. They don't have anything wrong with them. Often the first time it's given in humans, it is given in a special laboratory. There's 1 or 2 in Australia where they have hospital beds. They book in they sit there for 7 days. They're given 1 dose, and then they're monitored very carefully, have 1 million blood tests, safety tests, every side effects. So they start with 1 person, and then they start with another person, and they wait in between to make sure nothing terrible happens in between. And so these are, and they might start with a very small dose, and then they give a bigger dose, and then the next person gets a bigger dose. It's not the same person, and then they look at long-term side effects. And after they've found a dose that they think safe and they understand how it's metabolized, then they'll start giving multiple doses. So people might be given it for over a week or over a month. This normally involves a very small number of people who are generally young and otherwise healthy, who are doing it for altruistic reasons. The phase 2 study is the next study where we start testing it in someone who has the disease we're going to target. It's normally a very small study. In scleroderma, they might use 30 to 80 patients. It's mainly a safety study. It's a bit of a look at whether or not it works, but the main aim of this study is to see if it's safe and to see if it's having some sort of target effect in the disease or the processes. And again, looking how it's excreted, how long it stays in the body, how well it's absorbed. So it's often quite an intense study for the person participating, in that they often want to have lots of blood tests. Phase 3 studies are much bigger studies. These are the studies where we really look at whether or not it works. And phase 4 studies are more sort of long-term safety studies. So the drug's out, even sometimes on the market, and they're looking at long-term side effects over 1, 2, 3, 5 years. So as I said, the phase 2 studies are the early phase studies. We do see these in scleroderma. We've had some in the last year or two at our site. Main focus is on the actual safety and the appropriate dose. They might try 2 or 3 different doses up against placebo. They're often quite short. They can be 6, 12 months. And they'll often have quite frequent visits, every 2 weeks, every 4 weeks, because they're wanting safety bloods. And they're also often long visits because they want to measure, when you take the drug in the site, how well the drug's absorbed and how well it's cleared, because that helps us with knowing how often we need to give the drug. When a drug's given 3 times a day, it's because it's out of the body fairly quickly. If it's given once a day, it's because it stays in the body for a while. So sometimes people have to stay all day and have bloods every couple of hours. And they do often we're doing every couple of hours, a cannula's put in, so you're not having a stab in your arm every couple of hours. The phase 3 studies are the studies that are generally designed to work out if it works. Most of these studies are going to be what we call double-blind placebo-controlled studies. That means neither you nor the doctor who's running the trial knows what's going on. Not knows what's going on, knows who is on the active drug and who is on the inactive drug or placebo. Generally in scleroderma, they need 300 or 400 people enrolled in the trial to show that it works. And this is challenging because getting 300 or 400 people with early diffuse scleroderma is not easy if you've got a drug that you think works on skin. And often these are now massive multi-centre international trials with 200 or 300 sites around the world. They generally last at least a year. Often there's an open label, sometimes 2 years, and there's often an open label extension where everyone gets the active drug. Phase 4 studies are often very large open label studies looking at long-term safety that can go on for years, and they can also be used to compare 2 different treatments. Less common in scleroderma, but for example, in rheumatoid arthritis, there's all these new biologic drugs, and now they're starting to compare one against the other. So they might compare a TNF inhibitor against a JAK inhibitor in rheumatoid arthritis and look at the long-term safety in terms of cardiovascular risk, heart attack risk, or cancer risk or something. So they might go over 5, 10 years. Often the drug's already on the market or it's just coming onto the market, and these are very long studies. As we haven't found a lot of drugs that work in scleroderma yet, these are less, and you'll see less of these in scleroderma. So people often, we use this term, it's blind, it's single blind, it's double blind. And people often want to, I need to explain what that means. And people often want to know, why is it blinded? Why don't I know what I'm getting? The reason that we have blinding is to try and avoid bias. Many of the outcome measures in trials, not only do we try and have hard outcome measures like, a measurement of something, like for a lung disease, it might be the measuring your lung function tests. But in things like scleroderma, we often measuring your skin score. We also often have a lot of quality of life questionnaires for patients. What about your fatigue on this drug? What about your itch on this drug? What about this? What about that? Those sort of things are very subjective. And if I tell you you're on the active drug, there is a subconscious bias to sort of feel better on it. If I tell you you're on the placebo, there's probably a subconscious bias to be more negative. And that subconscious bias also exists for me. It's not out there in the open. You're not deliberately changing it. And that's why both of us will be blinded to know what's on the drug, who's on the drug. So neither you nor your doctor will know who's on what. And it's all very carefully done down in the pharmacy with randomization processes. Even the pharmacy does it. They get delivered boxes of pills. They're both the same. There's numbers on the boxes. It's very complex and very carefully done. At the end of the trial, you'll probably be told what you're on, but you won't necessarily be told halfway through the trial. So one common trial structure is that there's a double-blind period for a year, and then there's an open label extension where everyone gets onto the active drug. At the point that you move over onto the study, when you're in that point of moving over, there'll still be a whole lot of other people who are on the blinded phase, and they won't unblind the trial till the end of the trial. And that's, again, not to induce bias in me or the doctor who's assessing you, because if I know 4 of my patients, if it's a 1-to-1 ratio, it's half the patients are on placebo, half the patients are on an active drug, and then I know that the first 5 patients are on active drug, I'll guess the next couple might be on placebo. It won't necessarily be because of the way it's all randomized, but there is, again, this sort of bias to thinking that they're on it. And it is subconscious. It's not deliberate. And this is how we've learned that we've got to do these studies. In a blinded study, you can't choose. It's all just randomized. So the other thing is people sort of say, well, why do we need placebos? Why do we need, an inactive treatment up against the active treatment? We need it so that we can have a comparator either to background care or to standard of care or what the natural history of the disease is. As I've said, scleroderma is a complex disease, and lots of things look like they're working until you compare it to placebo, because people's skin was naturally going to get better at this phase. Here's a simple example. Over here on the right, I planted a little plant. I watered it with Wonder Growth, and a week later, here's my plant, and I thought, fabulous. Wonder Growth works. I'm going to water all my plants with Wonder Growth. Hang on. Over here, say I planted 2 plants at the same time. This one I just watered with water, not Wonder Growth, and at a week, the same result. So this is my placebo water. They both need water. We know it's not going to grow without water, but this Wonder Growth substance really didn't make any difference. And if I hadn't had that placebo, I would have told you Wonder Growth makes your plants grow faster and better, but maybe it doesn't. Maybe that was what was going to happen anyway. And that's why we need placebos. Here's a couple of examples of placebos in clinical trials. So the squares are the active treatment. The round dots are the people on placebo. So this was a trial of an oral drug in early diffuse scleroderma. And you can see that here, that they really, both groups look the same when you compared them. Particularly, how patients felt, their lung function tests, their skin score. The MRSS is their skin score. There's a little difference there, and you'll say, oh, there's a little difference, but that wasn't statistically significant. And if you look at the overall graphs, honestly, they look very similar. So without the placebo, we would have said, this is a great drug. Here's an example of where a placebo told us the drug works. So nintedanib is a drug that's now available for people with progressive interstitial lung disease, so people whose lungs are getting worse. Not everyone with interstitial lung disease, got a lot of side effects. But what you can see is that in this trial, and this was a huge trial, nearly 600 people involved all around the world, the patients who got nintedanib, their rate of change over 1 to 2 years over a year, their yearly rate of change, and this was a 2-3 year study, was smaller. So they lost less lung volume than the group here that were getting the placebo. And you can see that graphed out here. So the patients who had the placebo, they lost more lung volume than the patients who are on nintedanib. It didn't reverse their lungs, but it showed that it was working in slowing up the progression of their lung disease. And that was important to tell governments that they should spend $20,000 or more on this patient for people with scleroderma. They want to see value for money. So when we go to design our clinical trial, our trial of our new medication that we think might work in scleroderma, it's important we choose the right people for the trial. After all, you can't compare apples to oranges. Scleroderma, there's no point comparing someone with limited scleroderma with someone with diffuse scleroderma, someone with no lung disease to someone with a lot of lung disease, someone with late lung disease where their lungs aren't getting worse to someone with early lung disease. So this is why it is really important. Sure, you both got scleroderma, as the cartoon says on the right, we're both fruit, but my apple is never going to turn into an orange. Lots of drugs have been tried in scleroderma. You know, we're as frustrated as you all are. And there have been many clinical trials, not all of them in Australia, but very few things have been shown to make a difference. And we're trying to get better and better at designing the trials. And we often say, well, why don't these things that we thought were going to work? Well, it may be that they just truly don't work. What works in the lab doesn't always work in people. They might work, but we might have got the trial design wrong. We might have designed this very expensive trial in the wrong way. We might have the wrong endpoint. We might have got too many people with late disease, too many people with mild interstitial lung disease that weren't going to progress. I also think a lot of our trials are too short. Certainly with interstitial lung disease, it's not a rapidly progressive disease in scleroderma. With idiopathic so interstitial lung disease where there's no underlying autoimmune disease, certainly that is quite a rapidly progressive disease. And in nintedanib, the study I showed you before in idiopathic lung disease, the difference between placebo was much bigger. You know, the volume that the placebo lost was huge. And sometimes we're measuring the wrong things, which is why we're always looking at new activity indexes. A lot of the epidemiology, a lot of people have been analysing some of the big data sets that we've got around the world, the USTAR data set, ours data set, looking at, you know, what are the predictors of progression of lung disease. And, you know, we've understood more and more about this. Who does progress fast? How can we choose the right patients to show that these drugs work? It doesn't mean that in the end we won't be able to use them for the other people that don't fit the trial, but you've got to show in a short period of time that these drugs are working. Trials cost millions and millions of dollars, a clinical trial of a new drug. So you've got to make sure you've got the right people in it, because you don't want to spend a few $1 million and throw that drug out the door just because we didn't get the rapidly progressive people into it. So when you do a trial, you need an endpoint. You need something that you measure, because the long-term goal of a trial is to show the drug works, for everyone to feel better, but also to get governments to fund it. And governments are not going to fund something where there isn't a hard endpoint that we can measure and that they can be convinced it really works. So they're not going to be convinced by I feel better, by quality of life scores. And, you know, there are scores of quality of life. They're not convinced by that. And they want something hard. So what we choose as the hard endpoint, the primary endpoint, depends on what we think the drug's going to do. And we hope overall it'll help the overall disease. But generally they choose one endpoint and measure a whole lot of other endpoints as well. So if we think it's going to help the progression of fibrosis in the skin, they're going to choose people with diffuse skin disease, those that are going to get rapidly worse, nice early disease. And we use something called the Medsca skin score that has been validated. So that's been tested. We can reproduce it between doctors. There's very little variation between each doctor. And I'll explain how it's done. We also, for lung studies, it's a lot easier. It's got a bit of a harder endpoint, and we use something called the FVC, which is a measure of how big your lungs, how well they expand. We have had studies for digital ulcers, and so we use digital ulcer counts. We make sure people have had digital ulcers before, and then we recruit someone with a digital ulcer, and they do all sorts of things like photograph them, time to healing, measuring digital ulcer counts. And there's also severity scores for Raynaud's. There's Raynaud's diaries where you have to go away and record how many episodes of Raynaud's you have a day. But again, they're not very hard endpoints, the Raynaud's diaries, because a lot of it depends on whether you stayed inside. You know, yesterday was the coldest day. If you stayed home all day, which I hope you all did, then you probably didn't have as many attacks of Raynaud's as if you had to be working outside. So it is very complex. It's not very good endpoints. And that, again, makes it difficult to get governments to fund drugs because they don't like the endpoints. So where the primary trial is skin, we use this thing called the MedScar skin score. You generally have to go to a training course and you know, we use volunteers, we use people living with scleroderma as their volunteers, and basically at each site of all these 17 sites we rate the skin from 0 to 3. 3 is like rock hard skin that you can't pick up at all. 2 is moderately hard, no fine wrinkles. 1 is just a bit of mild thickening, and 0 is normal skin. And we go around the body pinching it all up. People say, well, why not my back? Why not this? It's just been shown to work. And if we have a trial that uses skin score as an endpoint, all the doctors have to pass an exam. All the doctors that are involved have to come to one site where we have half a dozen wonderful volunteers who've got scleroderma, and they're blindly rated just to make sure that they're rating it the same as a trained, experienced person. And this diagram is where we do it, but also this is a bit of a diagram of limited scleroderma. Limited scleroderma is only if you've got your hands and forearms, your lower legs and feet involved, your face. Diffuse is where you can have a little bit of the front of the neck, but diffuse is where you have the upper arms, thighs, tummy involved. We know that people, when skin's the primary aim, we know, I showed you that graph before, people who are going to have progression of their skin score. And what we want to see is that the drug stops progression of the skin score. We know that's the people with early diffuse disease. That's the group we've got to get to and test the drug in. Sometimes trials will include people in the 2 to 5 range if we've got evidence from their doctor that they've got new involvement recently or that their skin scores got worse recently. Some studies also set an upper limit for the skin score because there's been trials showing that if you've already got to a skin score of 30 or 31, you're probably not going to progress much more. As yet, we're not confident that we've got drugs that melt the skin score. So we're often looking for drugs that will stabilize it versus placebo. Well, they'll get worse. Some trials also want raised inflammation markers to enter the study, and that's because looking at these big registries, it does show that raised inflammation markers are an indicator people might have more active disease. That can be hard because a lot of patients with scleroderma, even early diffuse, do not have raised inflammatory markers, and that can put them out of a trial. And again, just a reminder, this is the group we want, and this is why, because if we get this group here, they're going to already improving, and, basically the treatment will look like the placebo group. Lung disease and scleroderma is also an area of great interest. We know it causes a huge amount of morbidity and mortality, but also we know that there are a large number of people with scleroderma, interstitial lung disease, particularly who don't have a lot of lung disease, whose lungs stay very stable. So if you look at this spaghetti plot on the right here, you'll see in there, here's one where their lungs go right down. Here's another one. But there's an awful lot of them where they're staying very stable. So what we're trying to do is choose these guys that are getting worse. And again, there's very good longitudinal data over some of the very big registry studies to suggest that the greatest loss in lung volumes happens in the first 4 to 5 years of scleroderma. Now, that doesn't mean that people with, that doesn't mean that people who've got 5-10 years of scleroderma aren't in trouble with their lungs, but they're tending to lose less amount each year. So it's harder to show that this drug is working out there. And again, when we've got a short time frame, you know, pharma hasn't got years to do these studies because it's costing them, you know, a few $100 million to do this study. They want to see it working fast. And so that's why they choose these strategies to enrich for a population that's going to be, oh, I can't work out why that keeps doing that, a strategy for people who are going to get worse. So some of the other predictors of worse lung disease are raised inflammatory markers, early loss, extent area in the CT scan that's 10 or 20% or more than that, early disease, and sometimes it includes the SCL70 antibodies that seem to be a predictor of lung disease. So to enter a trial, you know, where the primary endpoint is lung disease, you're going to have a CT scan that shows a certain amount of disease. And that CT scan will be read by a central vendor. So all the scans for everyone who wants to enter the trial will be sent there. They'll be done on a standard machine where they've got standardized settings and things. And again, generally it will be people within their first 4 or 5 years of scleroderma, because we think that's the group that are going to have the most rapid progression, where we've been able to really show that it's making a difference, and then we can expand it later to other groups. Sometimes they exclude people with very severe lung disease, again, because it's going to be harder to show it works. People who can't do lung function tests, sometimes people have a lot of trouble doing lung function tests, and people with very, very bad lungs have a lot of trouble doing it. Again, it doesn't mean later if we show it works, this won't be accessible, but we've got to try and get the right group of people so that we make sure we show it works, so that we do get these drugs. So are there any clinical trials for you? Actually, probably many people on the call today won't fit most of the current trials that are out there, because most of the trials that are out there are in this group of very early people with early diffuse scleroderma, rapidly progressive skin, or with early interstitial lung disease. And every trial has a very long list of inclusion-exclusion criteria. The inclusion criteria are the criteria we say you need to have all these things, and the exclusion criteria says you can't be in this trial if you've got this. When they're starting with a new drug, they don't want people who've got a whole lot of other problems, because anything that occurs during the trial is considered an adverse event. So, you know, if you've got a new drug and you're not 100% sure, you think it's mostly excluded by the kidneys or the liver, you don't want people whose kidneys don't work very well, because that might build up more and they might get more side effects. And, you know, it's going to be hard to manage. Or the same with bad liver. So generally, if you've got bad kidneys or liver, you're out of these early trials. Doesn't mean we can't use it later, but we just don't want it in the trial. They generally exclude people who've had cancer in the last 5 years, because, you know, that can make it hard to manage if the cancer comes back. Is it the drug? Is it not? Etc. They might be needing other treatments. They generally exclude people with a whole lot of other things. They're generally wanting people who've just got one illness, or, you know, illness plus a little bit, because everything that happens to you gets recorded as a side effect of the drug, even though it might not be. So there are always a long list of inclusion-exclusion criteria, which, you know, the doctors involved in the trials know, and, you know, we don't like recruiting people if we know they're just not going to meet the criteria and they go through all the tests and things. So if you hear a clinical trial, what should you think about? You think, oh, well, I have got early lung disease. I wonder if there's something for me. First of all is to ask about the inclusion-exclusion criteria, and do you fit the criteria? And I'll actually give you some websites where you can see scleroderma trials around the world. And these are the things that you would think about. Do I fit the criteria? Do they have the right type of disease? If it's a skin trial, do I have diffuse scleroderma? How long have I had disease for? The next thing to think about is what's involved for me. So how often will I have to go to the study centre? The study centre isn't going to be your local GPs or your local specialists. It's going to be a centre, a big hospital. How long will each visit take? It's not going to be the same as just coming into the office to see me. You're going to have blood tests, lung function tests, blood, and the blood tests are not just down the local pathology. They have to be done at the site. Sometimes you've got to have, blood tests before you take the medication and a few hours after. How long does the trial go on for? How long am I committing to being on this trial? We don't want people recruiting for a trial unless they genuinely think that they can manage to attend all the visits. You can always stop a trial at any stage. But generally, we want people who think that they can manage to do it all. You need to know what's known about this study treatment. Has it been used for a long time? Have only 20 people taken it? What do they think the side effects are? How many people have taken it? What's my alternatives? Can I stay on all the other drugs I'm on at the moment? Many trials in scleroderma now allow background therapy with mycophenolate or methotrexate. Some trials don't. Is the study placebo controlled? And what is the chances that I'll get active drug or placebo? Is it, a placebo controlled study for 12 months and then everyone gets active treatment? Or do I never get a chance at the active treatment? If I get worse, if my skin gets worse while I'm on the study drug or my lungs get worse on the study drug, sometimes we're allowed to add in other therapies and keep you on the trial. Sometimes we're not. And what tests do I need to do? What will I have to have another CAT scan of my lungs? What will I have to have done? How often will I have to have lung function tests? If you can't do lung function tests or don't like lung function tests and you're going to have them every month, maybe that's not the trial for you. So these are the important things to think about. And how is this different to if I just stayed on mycophenolate or standard of care, or what other options are available? So where are clinical trials held? These sort of clinical trials of new drugs are held in big public hospitals. Occasionally, there's a big private centre that runs them. Why is that? Everyone said, well, why can't my rheumatologist be involved in it? And the reason is that they're incredibly complex to run and set up and very complex to run. As I've said, if it's a skin outcome, then the doctor has to go off somewhere. They have to do a training course. These training courses are run by the big pharma. It's complex to run it all. It's complex. You need a lot of experience. Generally, the pharmaceutical company that sets it up, sets up one, maybe two places in each state. All the bloods that are done on the trial are taken at the site and they're sent overseas to overseas labs. Why overseas labs? Because they want the one lab around the world to do all the testing. They have to validate the machinery. They generally bring their own lung function machinery. That has to all be validated. The CT scans are done on one CT scanner. They've done dummy runs to test it, make sure it's all the same. They need staff to come in regularly and monitor all the data and check the data. They can't be running around to thousands of private practices. And the reality is, St Vincent's Melbourne is a big site where we've had over the years many clinical trials. Many of these trials, we just recruit 2 or 3 patients because of the inclusion-exclusion criteria, because of whether or not people commit. You're working full-time, you've got to come every 4 weeks. You might not want to commit to this. So what's the journey in a clinical trial? This is slightly backwards. First of all, it's the recruitment phase. So, someone in the clinic, or you might see an ad, you might like to talk to the site, talk to them about your disease, about the inclusion-exclusion criteria. You might be referred. Sometimes we get patients referred from other rheumatologists who know that we run trials. And generally, you'll be given someone will chat to you on the phone, then we might get you in and give you a detailed discussion. We'll give you what's called an informed consent form, which is often a 20-page document that goes through everything that's going to go on in the trial, all the information we know about possible side effects of the drug, and all the tests that you'll have to have, how long it will go for, etc. And then before anything gets done, you sign the consent form. You say, yep, I'm interested in this trial. I know what it's all about. I'm prepared to get involved in this trial. And then you'll go through what we call screening. And the screening might run over 4 or 5 weeks where you have a whole lot of tests. And it may be on some blood tests at the end, your liver function tests are up this week and you're not able to be included in this trial. Or you don't have enough on the CAT scan that we do. You don't have enough lung disease or something else, or the inflammatory markers aren't up anymore. And you might be excluded from the study and then you don't go on. Once you've passed the screening, the next then is randomization. This is all generally computer driven, and you're randomized either to the active treatment or which dose or to the placebo. Then starts the visits, and the visits will all be laid out. You might have to come every 2 weeks to start with, or every 4 weeks, just depends on the drug, how much we know about it, whether the drug has to be increased. Some drugs, the dose has to be increased over time, and you have to be calm and sane, and you have to have the majority of these visits at that trial centre. There are moves to try and decentralise trials with nurses going out from big companies and taking bloods and things out in the community. COVID has driven that a little bit, but it's not very common yet. Hopefully that will improve inclusion at the end of the day. But there's a lot of measures, things like lung function tests in scleroderma trials, skin scores, we can't do out in the community, but sometimes the safety visits. And then at the end of the trial, it's, well, what do we do now? Some trials allow you to stay on open label for a long period of time. Some trials don't. And then it's a discussion between you and the doctor. And that's something to think about too in the trial. Also, if they analyse all the data and there is an open label extension, and then they analyse all the data and they find it doesn't work, then they'll probably say, well, we're withdrawing this open label. What do you have to do if you're involved in a trial? It's really important you attend all the appointments. There's often very short windows, a week here and a week there. If you're planning a 3-month trip overseas, you're probably not appropriate for the trial. Anything that happens to you is considered a side effect. You get a cough, a runny nose, you get some antibiotics from the GP. We like it all recorded and you have to tell us about it. If you get admitted to hospital, we need to know about it. If you're going to start a new medication, you need to check with your trial team. They'll often tell you it's okay to have antibiotics or this or that, but it is important that there's no drug interactions and it's not a forbidden drug. You need to carry a card around that says you're on the drug. You rock into emergency, there's numbers they can ring so that, people can be contacted to make sure that it's not something to do with the clinical trial, or that the treatment they're going to give you for whatever it is isn't going to be an interaction with this medication. And the other important thing is, don't talk about this trial or how you're feeling on the trial on social media or advertise it to other people. It's really important we don't break biases. There's a lot of confidential information you'll be given about the mechanism of the drug, all of that. And you really are signing on to acting and behaving in a respectful manner to the trial team and to the company that owns the drug. Sometimes people, they think they're being helpful by telling everyone about it, but all advertising, and we are trying to increase the advertising to make sure that people know about trials, needs to be approved by the ethics committee of the hospital. And that's to make sure it's not misleading. And every now and then we see people get up on Facebook or on Instagram, all these things saying what a wonderful thing they're on and it works and all the rest of it. And that's just not what we want. So why would you want to get involved in clinical trials? Well, sometimes it's to access a new drug, a new treatment, but let's remember, they don't always work. Some people just want to contribute to medical research. They've got this rubbish disease called scleroderma and they just want to be involved. Even if it doesn't help them, they want to help the next group of people with the disease. And they want to improve their quality of life and other people's quality of life. Many people who are involved in trials tell you it's a very positive experience because you're monitored more intensely. Often progression of your disease is picked up earlier, even if the drug or treatment doesn't work. They often feel more engaged, that, they've got a nurse they can access all the time. They often get more education about the disease. So they actually enjoy it, even if it's time consuming and it doesn't actually result in a new treatment. So what are the pros and cons of it? It might give you access to a new treatment that might work. Certainly it might You do have often more intense monitoring of your side effects and your disease, and it does often help develop new treatments or teach us that that treatment doesn't work, so we stop developing that. But the cons are this treatment might not work. Just because we're telling you it works in a lab doesn't mean that it is going to work. So often people feel a bit upset when they're not able to be part of a clinical trial. But just remember, because they've met an exclusion criteria, or it's not exactly for their disease, or their disease is a bit late or a bit long. But don't forget, this drug might not even work. And it is time consuming. You know, this really is time consuming. Often our patients coming in for clinical trials are there for 3 or 4 hours just because of all the things that happen in a visit. It doesn't seem a lot, but just the organisation of it, going and having a booked lung function test, this, that, the other. And also, you know, sometimes people develop a side effect as a result of the treatment. And obviously we're intensely monitoring for that. And that's why you're having a lot more blood tests than you would normally to look out for this. But, you know, these are the downsides of being involved in it. So how can you find clinical trials in scleroderma? So there are websites. All pharma register their trials on this big website here, clinicaltrials.gov. This is an international website. In Australia, they're generally registered on the Australian clinicaltrials.gov.au, but not always. So clinicaltrials.gov is generally the best one. Hopefully, increasingly, we'll have websites such as, when you search these websites, they don't always work for scleroderma. You've often got to put in systemic sclerosis, particularly the Australian one. I was having a play yesterday and it seemed to need systemic sclerosis. The clinicaltrials.gov seemed to pick up scleroderma. You can talk to your doctor, talk to your rheumatologist. Sometimes they know about trials, but not always. Increasingly, we're trying to get websites to have some information about trials. And again, this has all got to be ethically approved by the ethics committee. So don't just do it yourself. Also the Australian Scleroderma Interest Group, we're updating our website, and hopefully we'll have a page about some of the trials that are around. But they may not be in your state. You know, there are a couple of states in Australia that do a lot of the trials, mainly Victoria and South Australia. There are a few coming to New South Wales. And if you don't have I spend, half a day a week unpaid doing these trials. Unless you've got a doctor that really is passionate about it and has got the time to do it and wants to do it, it's probably not going to be where you are. And that is unfortunate, but it's just part of how it is. So this is a study that is about to start in several sites around Australia. And it's a drug that Boehringer Ingelheim has. It is not a drug that's on the market. It's a drug that they think works on blood vessels and also on the fibrotic pathway. So they're really targeting people who've got relatively early scleroderma again, early diffuse scleroderma, as well as vascular problems. So not only do they need to have skin disease, but they also need vascular problems. To enter this study, you need to be 18 or older. That's generally a requirement in Australia. It's complex, but that's generally what you'll see. You need to have this diffuse scleroderma of less than 5 years. And this is one which has got a lot of complex inclusion-exclusion criteria. It does my head in slightly. So, diffuse scleroderma, again, you've got to have got some thickening at the time you enter on the upper arms, the thighs, or the tummy. And that means slightly stiff skin. You can't have limited scleroderma. You can't have scleroderma their first symptoms started more than 5 years ago. If you're between 2 and 5 years of scleroderma duration, you'll have to have demonstrated some progression of your skin thickness over the last 6 months. And you need to have have Raynaud's or digital ulcers in the past. And you need raised inflammation markers at the time you enter the trial. They're not particularly high, but they've got to be slightly up. If you've got bad kidney or liver, you won't be included in the trial. That'll be an exclusion criteria. If you've got very severe lung disease, or if you're on medication for pulmonary arterial hypertension, you'll be excluded. Some background treatments, such as rituximab, which is an infusion treatment in the last 6 months, you'll be excluded because that's got a very long action. You can be on mycophenolate or methotrexate or azathioprine to enter the trial, but the dose needs to have been stable for 4 months before the end of the trial. And again, this is so that you've got to a sort of set point that things are stable, then we're going to see the effect of the drug. So the study design of this drug, this one's a complex one too. There's a screening period for up to 5 weeks. Then there's a primary treatment of nearly 12 months. And then you'll go on to an extended treatment. This is placebo-controlled. It's 1-to-1, so half the people will be on placebo, half the people will be on active drug. The extended treatment period for up to 72 weeks, it'll be basically the study will stop when the last person into the study has had 48 weeks. And you're not going to get all these hundreds of people they need around the world all starting on the one day. And then there's always a follow-up period where you go off drug for 4 weeks. It's possible that as time goes by, they might put an open label in. It's possible they'll do an analysis halfway through and say, actually, this isn't working, and they'll pull the study. That can happen. Or if they see suddenly, they're constantly monitoring side effects, and if they suddenly see abnormal side effects they didn't expect, they may pull the study. You'd have to come to the study site about 14 times, or probably more, like 20 times if you're if you're recruited early. At the start, you have to come every 2 weeks for the first month because the dose is gradually increased, and then every 4 weeks for 2 months. And then after the first 2 months, it's every 6 weeks. So that's coming down and visiting the study site. Once you go into extended treatment period, it does move out to 3 monthly, which makes it easier. But you're committing to a 12-month period. There are little windows, but, you know, it's generally only a week either side. It's a tablet. You take it 3 times a day. And some people will be on placebo, and none of us will know what you're on till the end of the study. So this is a list of the participating hospitals, and Scleroderma Australia website will have the ways you can contact these hospitals if you think you meet the criteria, if you want to talk to someone more about it. So not all clinical trials are about drugs, fortunately. My focus has been on drugs today. Trials might be more about trying to diagnose something earlier, lifestyle interventions. Here's a couple of other trials. These are only available at St Vincent's Hospital, Melbourne. You have to be in Melbourne to be, or in Victoria to be on them. This is a study looking at SIBO, how to diagnose SIBO. So they're looking for people with scleroderma. Doesn't matter how long you've had scleroderma for, doesn't matter if you've got limited or diffuse scleroderma. And they're looking for people to try and work out what tests do we need to diagnose SIBO and a particular treatment that might work for SIBO. So we want people who haven't necessarily been diagnosed, that have got bloating, diarrhea. You can have limited to scleroderma, diffuse scleroderma. It'll involve a whole lot of tests, a whole lot of visits, including breath tests, an MRI of your small bowel, ultrasound, and it may include a gastroscopy or colonoscopy, depending on some of your results and depending on whether you've had one or not. And then they may give you a special antibiotic if they think you've got SIBO. So it's a good way to try and sort out what these symptoms are due to. This has been done, one of our PhD students, Alana Quinlivan, and that's Alana's email if you're interested. But it is only going to be at St Vincent's, Melbourne. And this is a study looking at arrhythmias, so funny heart rates. Again, it is only at St Vincent's, Melbourne, and the Baker Institute in Melbourne, and they're looking for people who've got scleroderma without any history of heart disease, and they're wanting to see how common it is to have funny heartbeats. So it involves 3 to 4 study visits over 12 months, an ultrasound of your heart, an MRI of your heart, a Holter monitor, and exercise tests and blood tests. So they're looking also for control groups, so that's family and friends who might also like to have all these tests and be a control group. And Jessica Fairley is our PhD student who's running this only in Victoria. Jessica's due to have a baby any day now. So if you contact her, don't worry if you don't hear for a month or two from her. And this study is up and running now. All right, so that's my end of my talk. I will escape the screen and go back to this screen and maybe stop sharing. All right.
Brilliant. Thank you, Wendy. That was a lot of information and very insightful, because I know that when you see a clinical trial, a lot of people, how come I didn't get into that or whatever, you know, so there is lots of nuances to it.
Yeah, that's why I wanted to try and explain this all. It's not that we don't love you all. We love you all and we care about you all. We really care about you all. And we know that people have got a huge amount of dysfunction, you know, discomfort, disability. But what we've learned from the past is if we don't get the trial right, we're binning this thing that we've spent $1 million on, or a few $1 million on, and it won't be any use. Once we get it, then we can sometimes extend it.
Okay.
So